My name is Mitchell Warren. I direct AVAC and delighted to welcome everybody to this webinar just past World AIDS Day that commemorates both so many things over 43 years now of this epidemic of great loss, but at the same time, great progress in some areas, continued sluggishness in others, and opportunities certainly in many respects with what we all need to do to really translate great science into impact. And that's perhaps the best type of introduction to this call today. I think we all are well aware that of 2024, at least in HIV science, amongst the biggest news, if not the biggest news, were the results of the two efficacy trials of injectable lenacapavir for PrEP. We are going to very quickly, I'm going to introduce a friend and colleague from Gilead Sciences who led that program. You can see the agenda now here, who will walk us through those results, some of which were published a few months ago, some of which were just published last week. We will then open it up for a Q&A, and I know there are other people on the line from Gilead who will be part of that Q&A. And then we are going to preserve some few minutes at the end to make sure everyone's aware of a new document that AVAC just launched yesterday that really hopes to accelerate action by all of us, product developers, funders, policymakers, program implementers, you name it, across the board to make sure that we don't squander this opportunity as we sadly have, I believe, squandered so much in the last decade with respect to PrEP. We're recording this call and it will be shared with all of you in an email in case you have to drop off early or if you want to listen to it a second time, you are most welcome. And we will also be posting it on the website. I encourage you to use the Q&A feature to ask any questions as they emerge. I will be doing my best to field those as they come in, and then hopefully we'll have time to open up the line as well, and we'll look for hands and go from there. That is the run of show. And again, I want to thank everybody for making time. I know it's toward the end of the year, people thinking about holidays and hopefully the festive season, but this is too important to I think our collective conversations about HIV and AIDS to not have a chance to really unpack the data and planning ahead. So without further ado, I'm delighted to welcome Mopli Das, who has been at Gilead Sciences for several years now, and most recently has been leading the Purpose program, which I know she will describe in its entirety. She oversees other aspects in clinical development at Gilead around HIV, but excited to have her here as the architect and implementer with many investigators, both in Gilead and around the world, in both the Purpose 1 and Purpose 2 trials to make sure that everyone has the data at hand and knows where we then need to go with that data. So Mopli, with that, I'll turn it over to you.
Thank you, Mitchell, for that lovely introduction. And I'm going to get started and just dive in because I know we want to get started. Okay. All right. Here are some disclaimers. I want to make very clear that lenacapavir is an investigational drug that we are still studying, and this is a non-promotional presentation, and we are here at your invitation to share our data. First thing I want to say is thank you. Thank you to AVAC in particular for all of the work that you've done to work with advocates over the last decade to help educate and train people to engage in clinical trials. We could not have done this program without AVAC and the larger community of HIV prevention advocates. We wanted to highlight for you in both the papers, we are delighted to have thanked the advocacy groups that we worked with and engaged for both studies. So in Purpose 1, we had a global community advisory and accountability group. And in Purpose 2, we had our GCAG. Both of these groups have published on their experience and shared their knowledge and advice and and critiques that they have given us about the program. And we really want to just start with gratitude to all of you. And you'll see the influence the advocates had on shaping this program throughout my presentation, hopefully. Okay, so many of you have seen me present before, and we love to talk about the innovation and the science in the program, the novel counterfactual design. I won't focus on that today. We'll talk about our new product, which is a twice-yearly subcutaneous injectable, lenacapavir. We also talk about how we feel we've changed and innovated in equity along with all of you, changing hearts, minds, and policies to allow some of the innovation in our program to take place. And we also want to talk about sharing the responsibility with all of you to enable broad access globally for this product when it's approved. Okay, so the first trial I'll be talking about is the Purpose 1 trial. And the second trial I'll be talking about is the Purpose 2 trial. And for those of you who are not aware, we obviously pursued these trials in the populations that we studied because of the disproportionate rates of HIV incidence in the communities of cisgender women and also in cisgender gay, bisexual, and other men, transgender women, transgender men, and gender non-binary individuals who have sex with partners assigned male at birth. The uptake of PrEP remains suboptimal in these groups, as Mitchell alluded to in his introduction, and we wanted to see if a different product with long-acting properties that was subcutaneous might help solve that uptake and adherence challenge that we've seen in the PrEP space. So this is a whole program. I'm going to talk to you about the results from Purpose 1 and Purpose 2, and I'll tell you a little bit about Purpose 3, 4, and 5, which are in the field at the moment. So Purpose 3 and 4, as you know, are collaborations with NIH, NIAID, and NIDA, and the HIV Prevention Trials Network. Purpose 3 is taking place in US cisgender women, so we can have data in US women because their incidence is too low to have been included in Purpose 1. In Purpose 4, that's the study in US people who inject drugs. Again, people who inject drugs were not excluded from the pivotal trials, phase 3 trials, Purpose 1 and 2. However, we wanted a program focused specifically on people who inject drugs with extra supports and outcomes evaluated to understand what's happening with people who inject drugs who receive lenacapavir. And then we also have Purpose 5, which is taking place in France and the UK to help us answer the question about annual persistence or adherence over 1 year and how that differs between long-acting and daily oral products. And what's really exciting today is, if we have time, I have a little bit of data about the Purpose 5 endpoint using the Purpose 1 data. And as I've alluded to before, we couldn't have done this program. The tree and the branches could not have blossomed without all of the critical aspects of our pillars of health equity, as noted below. Okay, so these are our foundation pillars of purpose of health equity. So this includes partnerships, voice of the people, which you all here are critically important for, person-centric design, and our focus on including those people who have been historically excluded and underrepresented in PrEP clinical trials, so we can have data in all the people who are most disproportionately affected by HIV when we launch the product. And we wanted to be very clear that we were intentional in our innovation to build towards health equity, so we didn't further exacerbate the existing disparities in the HIV epidemic. So some of you are on this call or in this picture. I see Gerard here. And this was our first meeting that we had before we even started writing the protocol for Purpose 1. And this took place in Kigali in Rwanda in 2019. And you can see some quotes from people who attended. The representatives here included community advocates as well as policy folks and investigators that eventually worked with us on the clinical trial, and some of our advocates who worked with us on the community advisory group. And this meeting was really critical. And just the next week, we had a meeting with AVAC, organized by AVAC to also discuss Purpose One that following week in December. It was a busy week of travel for many of us. So this timeline just really highlights our early committed and continued consistent stakeholder engagement in the Purpose program that started with those meetings in 2019. The AVAC ones are on the bottom here, and then continued over time. And due to the pandemic, we did more Zoom meetings. But once we were able to travel, we started bringing folks to us and visiting people all around the world. And having meetings with our investigators and our community regionally, globally, so we could most robustly implement the trial according to the principles of good participatory practice. Okay, so speaking of those GPP principles, these are all the things we've done, and I want to quickly go through this presentation on this part because many of you are familiar with our history, and we've actually published manuscripts and given several presentations led by our GCAGs about this story. But these are some of the things that our community stakeholders and our GCAGs and many of you told us to do, and we did implement every little bubble on this slide. We really focused on person-centric design. We included adolescents for the first time ever in adult pivotal trials, so 16 and 17-year-olds in our adult pivotal trials. Due to the feedback from that first meeting in Kigali, we really tried to change hearts and minds and include pregnant people and lactating people in our program and also study them to make sure that we have all the data to allow people to use PrEP with the most knowledge in some of the times that are highest risk for people for HIV acquisition, which includes pregnancy. And I just want to shout out the many advocates on this call who were involved in these manifestos and this advocacy to support us in doing this. And people often ask us, how did we do it? And one of the things we did was share the science. So everybody internally, externally, at regulatory agencies, at the ethics committees were familiar with the data so they could help us support our policies. I wanted to highlight the impact and WHO toolkit, which is supporting this for other people who want to follow our policies. There's a really robust toolkit with a website that can help you do this in upcoming trials. I just want to highlight, this wasn't just in Purpose One. We included pregnant and lactating people with and people with childbearing potential throughout all of our programs. Okay. And this was a presentation in Munich about our experience of our community. And I won't read all these quotes, but we just wanted to highlight their reflections from working with us on this important program. And I can send that deck to you later. And Purpose Two, also, our first publication in Purpose Two was actually about our GPP strategy. And that was highlighted in this publication from 2022. Okay, and in Purpose Two, one of the highlights, really following the No Data, No More trans manifesto, was the inclusion intentionally of transgender women, as well as really intentional inclusion of trans men and non-binary people in our trial. And we were the first PrEP trial to include trans men and non-binary people. Okay. And one of the other things that we were told, and we really appreciated your support about, was updating our website to be inclusive and representative. And this really allowed us to recruit and engage with the communities that we wanted to enroll in our study. And we tailored those locally. This was from a presentation given at R4P in Peru. And for those of you who weren't there, this presentation was given by Jorge Gallardo, and Gallardo Cartagena, and he included some beautiful photos from his site that they developed with the resources to recruit in Lima. And okay, now I'm going to get into the results. I just wanted to do that first piece to really thank all of you and the larger global community for all of your support in these programs. We really couldn't have done it without you. Okay, here's the Purpose One study design. As you know, we had the incidence cohort at the beginning to evaluate the background incidence or the counterfactual. What this cohort did was, with recency testing and the RETA algorithm, allow us to figure out what the background incidence was and use that as a comparator arm. In this study, we had two products that we were studying, lenacapavir and FTAF. Everyone got a pill and everyone got an injection. Some people got a small pill, some people got a big pill, and no, everybody was blinded. And when 50% of people were through week 52, we did an interim analysis, and we were only able to stop the study if lenacapavir, the incidence on lenacapavir, was significantly less than the incidence in the background incidence cohort, and it was superior to Truvada, which scientifically means that it was much lower than the incidence on Truvada. So we achieved both those outcomes in Purpose One and Purpose Two, which, as you know, had a very similar design. It excluded Descovy or FTAF because we already knew the data of FTAF in these populations from the Discover trial. So here is the global distribution of participants in Purpose One. So more people were from South Africa, and we had more sites in South Africa, which makes sense because South Africa has the highest incidence and prevalence of HIV globally. And here we had the distribution in Purpose Two. And you can see we had very diverse representation across 5 continents in these trials. Here is the distribution of participants by race, ethnicity, and gender identity. 67% of the participants were non-white. Similarly, approximately 63% were Hispanic or Latina, and 22% of participants were transgender or gender diverse. In comparison with prior PrEP trials, we are very pleased to share with you that we had an extremely globally, racially, ethnically, and gender diverse trial. We were the first trial to intentionally include trans men and gender non-binary people, as I mentioned, and you can see our results and percentages across the bottom compared to other pivotal PrEP trials here. Okay. So with respect to the baseline characteristics in both trials, the participants' baseline characteristics were well balanced across all the arms. Things to point out, in Purpose One, the age group was 16 to 25, and we were very happy to have over 110, I think it was 124 adolescents, age 16 to 17 years old. In Purpose Two, the age range was 17 to 74. And a third of our participants were between 16 and 25. So we have a really nice chunk of people in the young and adolescent and young people category. We had the diversity I told you about in Purpose Two. Given the geographic distribution of Purpose One, it's no surprise that almost 100% of participants were of Black race. Lots of STIs when screened and laboratorily diagnosed at baseline in both studies. And in Purpose One, we had quite a high rate of prior HIV testing, but not many people had ever used PrEP in their lives. So if you think about 16 to 25-year-olds, almost 80% of them had ever had an HIV test, which I think is a testament to public health testing in these locations. For Purpose Two, an important factor to point out is between a fifth and a fourth of folks reported self-use of stimulants and other drugs with sex or chemsex. Okay, with Purpose One, there were, as many people have heard or maybe saw in Munich or in the newspapers, there were 0 infections in the lenacapavir arm, and the incidence on FTAF and FTDF was very similar to the background incidence, indicating that not a lot of efficacy here in the FTAF and Truvada arms, and that was driven by low adherence, which I will show you in the next slide. In Purpose Two, we had 2 infections for over the similar amount of person time of 1,900 person years, and a very similar background incidence. And we had a lower incidence in Truvada than the background incidence arm. And you can see the median follow-up duration on the slide below. Okay. in terms of the efficacy results, in Purpose 1, we had 100% efficacy, which meant 100% risk reduction or reducing HIV incidence on the study product of LEN compared to that background HIV incidence estimate. And similarly for Purpose 2, we had a 96% reduction because of those two infections. So compared to the background HIV incidence. And as I mentioned before, there was no difference in the FTAF incidence and the background incidence. Okay, in Purpose 1, our secondary analysis in both studies was comparing to Truvada. So here we're using the term superiority in the scientific essence, which meant that it was statistically significantly much lower than the incidence in the Truvada arm. So LEN reduced HIV infections by 100% in Purpose 1 compared to Truvada, and 89% in Purpose 2 compared to Truvada. There was no difference, again, between FTAF and the Truvada arms. Okay, so why did this happen? So our explanation here really of the difference in efficacy is driven by the difference in adherence to the injections compared to the oral products. In Purpose 1, injections for LEN were on time 91% of the time in week 26 and 94% at week 52. And here we have our usual representation of adherence. Green means more than 4 doses per week, so very few people were taking more than 4 doses per week. Yellow is 2 to 3 doses per week, and red is less than 2 doses per week. So most of the women in Purpose 1 started out, half were taking less than 2 doses per week, and that continued to increase over time. In contrast, in Purpose 2, LEN injection adherence was similar at 90% at week 26 and 93% at week 52. However, the adherence, you see the green bars are a lot higher in Purpose 2. So adherence started out high in the 80s and decreased somewhat, but you see a lot more green on the Purpose 2 side than the Purpose 1 side. And this was due to the difference in the pill-taking behavior between the two populations. In comparison, for those of you who are familiar with our Discover trial, just wanted to pop this up to remind you this doesn't mean that Descovy or Truvada are not good drugs for PrEP. If they are taken, they have incredibly high efficacy, as was demonstrated in the Discover trial. So in FTAF and Truvada, we had very, very high rates of adherence in Discover and concomitant very high rates of efficacy in both arms. Okay. So with respect to Purpose 1, people may ask, but what about Descovy? You were trying to figure out if Descovy or FTAF worked in cisgender women. And to answer that question, we looked specifically at the people who got HIV and their matched controls. And we did this thing called a case control analysis. And what this showed is that there was a 90% lower odds of getting HIV if you took your pills. So more people took their pills in their control and fewer people took their pills, their Descovy. But if you did take your Descovy, it was highly effective in reducing your risk for HIV infection. Okay, in Purpose 2, we had 2 infections. So I want to take you through our understanding of the attributes of what happened with those 2 people who acquired HIV. And I'm going to stop right here and say, because there were only 2 people, there's a lot of information that the sites know about these participants and that Gilead knows and our investigators know that we are not sharing publicly because there's only 2 people. So the amount of data that I'm going to give you is limited about their demographics and their personal private health information because we do not want to reveal too many details since there's only 2 people. So know there's going to be a lot of questions about detailed aspects of the 2 people, and I'm going to tell you what we think is acceptable to share according to GCP and privacy rules and regulations. Okay, what you're looking at here is distribution of 10% of the participants who were in a randomly pre-specified cohort that we chose before we did the study. So we chose 10% of participants to do lenacapavir plasma PK on. And The dark line in the middle is the median concentrations of lenacapavir. Each dot is a concentration, and then the dotted lines are the 5th and 95th percentiles. So this is the LEN PK in 10% of the cohort, which is the representative PK. And it's also important to note here that this PK is similar to the PK that we observed in the LEN for treatment studies and in our other studies with lenacapavir in our development program. Now, let's talk about the first case. The first participant was a trans woman who had latent syphilis diagnosed and treated at baseline. She engaged in transactional sex, and she was diagnosed with HIV at week 13 with absolutely standard HIV diagnosis, standard testing. So she had a rapid that was positive when she came in for her visit. Her central lab rapid, which was sent to the lab and came back in a couple of days, was also positive. She had signs of HIV-1 infection in the confirmatory HIV differentiation assay. It was intermediate, but it was confirmed later because her qualitative and quantitative RNAs were positive for HIV-1, and her quantitative RNA was 934,000. And here you see her lenacapavir concentrations with respect to the rest of the cohort. Now, here's participant B. Participant B was a cis gay man who had rectal chlamydia diagnosed and treated at screening. He was diagnosed at week 26 with standard HIV testing again. In this case, he had a rapid negative test, and his labs that were sent to the central lab were all positive for HIV-1. He had a positive antigen antibody test at the central lab, which came back in a few days. Then he had an intermediate positive HIV-1 antibody that was later confirmed by his qualitative and quantitative, which came back a few days later, and his quantitative RNA was 14,000. Here you see his lenacapavir concentrations, and you can see that it is within the range of what is expected and all the other little dots of lenacapavir concentrations. So both of these participants were diagnosed with standard serologic HIV testing at weeks 13 and weeks 26, respectively. There was no evidence of delayed HIV diagnosis, which is another way to say Levi syndrome. We did not have any evidence of Levi syndrome in these two cases. And both cases had LEN concentrations which were within the range of the 10% random subset of participants and similar to prior studies. And both participants had acquired N74D capsid mutation at diagnosis, at HIV diagnosis, that they did not have previously. This suggests that this resistance mutation was acquired as the person acquired HIV infection. The capsid mutation was generated. There's no evidence that N74D is circulating, and this was not therefore due to transmitted capsid resistance, which does not happen due to various factors, including the fact that there is not this mutation circulating in the community. Okay, moving on to pregnancies. In Purpose 1, we had 510 pregnancies. We continue to follow these participants. Not everyone had delivered by the time we had the interim data cut. And if you look at the rates of interrupted pregnancies, especially of spontaneous miscarriage rates, these are very consistent, in fact, lower than reported in terms of the expected rates in the geographic locations where we did the study. And we're going to talk about safety now. These graphs do not include injection site reactions, which I'm going to talk about in the next slide. Here we have Purpose 1 and Purpose 2 safety. On the left-hand side, Purpose 1, and you remember both the trials were randomized 2 to 2 to 1 in Purpose 1 and 2 to in Purpose 2. You can see that most of the adverse events were low grade, and there were very few AEs leading to discontinuation of the drugs seen here. You can also see that most of the common adverse events were headache, UTI, STIs, upper respiratory infections, and nausea and vomiting. One thing I do want to highlight to you that in Purpose 1, there was a lower rate of nausea and vomiting in LEN numerically than in FTAF and Truvada. And that was really one of the only things that was different between the arms, and this was, numerically different. but notable. That's something that we're continuing to follow. Here in Purpose 2, again, the AEs were low grade, low rate of AEs leading to discontinuation. Most of the AEs or adverse events in this study were really STIs, along with other common maladies like diarrhea, headache, influenza, and nausea. Nausea was also slightly lower in the LEN arm in Purpose 2. Another important difference in Purpose 2 was there was a little bit lower renal function in the Truvada arm, just 5 mils per minute in the eGFR, which is not a big deal clinically, but that's consistent with the profile of Truvada and reflects the changes due to Truvada on kidney function. The LEN kidney function remained stable. So that's the general safety. And now I'm going to move into injection site reactions. Before I talk about the ISRs, I want to really explain how lenacapavir works. Lenacapavir is a drug depot drug. What that means is it's injected through the skin into the sub-Q space, which in most people has some amount of fat. You got skin, fat, muscle. That's the way to think about it. Something like cabotegravir or your flu shot goes into your muscle. So that goes through the skin, through the fat, into the muscle. That's an IM injection. Here, we're talking about subcutaneous injection, which means under the skin, subcutaneous. So LEN gets injected there and it forms a little depot. And that depot contains the drug, and that's the mechanism of action. That's how the drug works. It sits there in the sub-Q space and it elutes out drug over time. Some people's bodies make a foreign body reaction or a granulomatous response to this drug depot, and some people don't. And depending on how much fat is in the sub-Q space and the person's body habitus and whether they make a response or not, some people may have palpable nodules where you can feel the bump under the skin. These are usually palpable but not visible. And as the drug elutes over time and that foreign body reaction resolves or reduces or absorbs, the nodules decrease usually before the next injection. But the existence of the nodule really reflects the drug depot because that's actual drug under your skin. Now, if that were injected much lower, for example, in the muscle, it's hard to feel because it's under so many more layers. Now, what's important in both Purpose 1 and Purpose 2 is the reports of the injection site reactions. What we're representing in these graphs are the 3 most common injection site reactions. So for Purpose 1, the most common was nodule, then pain and swelling. In Purpose 2, that was nodule, pain, and erythema. Erythema was not as commonly seen in Purpose 1 due to the racial makeup of the population. Now, on each of these graphs, if you look at each little bit separated by the dotted lines, what you have is the LEN results and the placebo results. So for people who receive placebo injections, which included PEG, you'll know that there was a real similarity in the pain, swelling, erythema between LEN and the placebo arm. The way to read these graphs is green means no injection site reaction at all. Yellow means a mild reaction, which is grade 1, and red means a moderate reaction or grade 2. So for pain, especially in Purpose 2, pain and erythema are quite similar between the two arms. Similarly, in Purpose 1, swelling, very similar between the active drug arm and the placebo injection, suggesting that these injection site reactions are really driven by the fact that you're getting an injection rather than the drug product. Now, with the nodules, that's different. There's more grade 1 and grade 2 nodules in the LEN arms, which makes sense because they got active product. But the people in the placebo arms also experienced some nodules. And that's due to the fact that they had a drug depot, they had a depot of the placebo injection material or PEG. Every one of these decreased over time. On the bottom, the 3 bars are the baseline, the week 26, and the week 52. And you might be asking, why did these decrease over time? And we think the reason for that is really threefold. One, we learned better how to give the injections during the study. And when I say we, I mean Gilead, the study team, the investigators, and the site staff. As we got more experience with lenacapavir in the Purpose program, which, the whole program has over 9,000 people in it, compared to maybe 100 people in the Capella study, we learned more about how to give these injections, so we had improved technique over time. The second thing is we also instituted pain medication measures because we learned better that the immediate pain, the initial pain from the injections, is better treated with ice. And so we started doing ice and cold compresses during the study, and that may have also affected the experience of pain. The other thing is this trend has been noted in other injectable studies, including of lenacapavir, that people report fewer injection site reactions over time. And that's probably due to the first two factors I mentioned, the improved experience, the improved pain mitigation, and the ability, the knowledge of what's happening and the experience. So people may be more used to what happens with an injection over time. Okay. So, in summary, we believe twice yearly lenacapavir in the clinical trials was highly efficacious, safe, and well tolerated. And we demonstrated these results in the most globally, racially, ethnically, age, and gender diverse phase 3 program of clinical trials done to date. And all participants are now being offered open label LEN. Before I go into the next presentation, Mitchell, do you want me to stop now and take questions about Purpose 1 and 2, or do you want me to, what do you want me to do? I can't hear you. I think you're on mute.
So sorry, Mopli. We are quite over, and there are a lot of
Okay,
questions already. I think in the interest of time, what I'm going to suggest, there are a few questions that have already come in about Purpose 1 and 2, the data you've presented, and a lot of questions about access, which I know is a hot topic for everybody. I think this next set is a persistence in terms of this. I think I might suggest we table that for now. Let's do Purpose 1 and 2, and I do want to make sure we get to this so that we really can dig into some of these questions. And I know that you are not alone and have colleagues on the line from Gilead, but maybe I'll start with some Purpose 1 and 2 questions that have come in, and
sure.
you might have seen the chat. Questions certainly about the two infections. And I think actually, let me go back. Tracy Swan, I think, asked it really nicely with respect to. Oh gosh, hold on. Um, I had it here. Tracy, where did you go? Um, but basically, to unpack a bit more about those two infections and what it might mean, and something to the effect of, and Tracy, most transmission happens from someone with acute HIV because viral load is so high. What is the viral load threshold that you might be using for testing LEN preclinically? And if it's not high enough, people will still be vulnerable to infection despite LEN use. This might be happening because of high HIV viral load, not low LEN concentrations. So can you help unpack those two infections? Because I think the other question from Ida in Zambia related to how do you explain the difference between the 100% and the 96% in the two studies? So maybe we'll start with that.
Great. Yeah. So in this clinical trial, we didn't have any data about the viral load of the people that the participants were having sex with because they weren't participants in our study, just the people in our study. So we obviously transmission happens from partners and viremic partners, but those partners were not in our study. So I can't tell you anything about the viral load of those partners of the two participants. What I can say is there is nothing different about the sexual behaviors of these two participants or their attributes than everybody else in the studies in Purpose 2. Of the over 2,300 people in the Purpose 2 LEN study, 99.9% of them didn't have HIV infection. And if you look at the New England Journal article, you'll see a lot of them were having chemsex. A lot of people were having behaviors with multiple partners, lots and lots of continued STIs, high incidence of STIs in the study. So there's really nothing different about these two specific cases than the other people. And I wouldn't take any of the attributes that I've told you about these two people. Like one was a trans woman, one was a cis gay man, or anything about their information that we have shared and say it's because of that that LEN didn't work, because there were 99.9% other people who also had those similar attributes who did not get HIV infection. And what was the second part of the question? Or did I answer it?
Oh, Tracy,
Tracy, I didn't answer. I see you shaking your head. Can you ask me what you want to, what again?
Sure. Thanks, Mitchell and Mapali both. The question is not what kind of sex people had or how many partners they had or what their partner's viral load is. So what's the amount of viral load that you tested LEN
Oh,
against?
sure. Yeah.
Because it
Okay.
might be about how much LEN someone has in their
It's about the
plasma,
virus. Yeah.
but how
Okay.
much virus the partner.
So,
Thank you.
so thank you for clarifying. I forgot that part of the question. Um, the viral load cutoff was less than 20 copies. So we used a very sensitive assay. So it was detectable less than 20 copies. It's the most sensitive assay in the field. We also did a special research assay called a single copy assay that looks down a single copy assay. So we're very confident that neither of these people had HIV at baseline or prior to their diagnosis in the study.
No, I'm actually not talking about the participants or the low end of viral load in a person. When LEN was tested preclinically,
Oh,
what was the threshold of HIV viral load? Like, did you look at it as affect the IC50
yes,
against 1 million copies, 10 million
yes,
copies?
yes.
Thank you
We
so much.
looked, yes, I'm sorry. I didn't realize you were asking about something not from our study. So preclinically, LEN was tested in the test tube and in phase 1B studies with people with a wide range of viral load, and in the test tube with an incredibly wide range of viral load from low to really, 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 really high. Okay.
I'm gonna, obviously, there's more clearly to
Oh, I
know
just
about.
looked at the chat. Oh my God, guys, sorry. I
No,
didn't
no.
see this coming.
I think let's leave that one there. I think there's more to follow up with on those two, but I definitely want to get to other things. Let me do two other quick things about data before we move on to the range of other questions related to access. And Lysias Zemba from WHO is asking, are the AEs disaggregated by population group and country? And did you see any differences in intervention group between the two studies and between different populations?
I highlighted during the AE presentation the only numerical differences we noted. So for purpose one, that was lower nausea and vomiting in LEN compared to oral PrEP. And in purpose two, a slightly lower nausea, but definitely less impact on kidney function. There was a small impact on kidney function in the Truvada arm and no impact in the LEN arm. Those are the only two differences. We have certainly looked at safety by a huge number of different subgroups and have done different analyses. We have not presented that yet, but what I will say is there's no group with any differences in safety that we have found.
Great. Another question from Manju Chitani, a colleague at AVAC. Any participants using LEN while breastfeeding? And if so,
Yes,
are they being followed and
yes.
monitored?
So in purpose one, if you got so first of all, in purpose one, you weren't required to use contraception, but if you wanted not to get pregnant, we gave you contraception free of charge. So there were a lot of pregnancies from people who desired pregnancy. And if people continued in their pregnancy, had their babies, and breastfed, what would happen in the study is once we discovered that somebody was pregnant, we did a informed consent process. And we talked about all the pros and cons, the benefits and risks of continuing on study product on the study. And remember, this was all blinded. And for the women who chose to consent to continue, we, and this is women because it's purpose one, all cisgender women. For those women, we followed them through their pregnancy and during lactation. And they also consented to have one lab test done on their baby for LEN concentrations. So we understand LEN concentrations during all 3 trimesters, postpartum, during breastfeeding, and in babies. We have not shared this data publicly, but there are no safety signals that we are concerned about, and we're going to do a deep dive on that data when we have the full data set of information about pregnancy coming up next year.
Great. And also, I know we are already with only a quarter of an hour left, but I'm hoping people will stay as long as they can, certainly for an extra 15 minutes, hopefully. One other quick question, Mopley, about data, and I'm going to parrot both Robin Eagle from USAID and Heather Ingold from WHO about additional data. Robin asking, when will qualitative data be available about participant experiences, and both with respect to pills, but also with respect to injectables? And Heather's asking, when will PK data for pregnancy and breastfeeding be available? So can you give us a sense of when additional data will come?
Coming to a conference next year near you very soon. So we're working on writing all of that up, and we'll be presenting it all next year. I already answered the pregnancy question. We're going to be waiting for everybody to have their babies. So we have a full data set of pregnancy, and for all the PK questions, whether it's gender affirming hormones, hormonal contraception, different phases in pregnancy, all coming next year, qualitative data and quantitative participant reported outcomes in everyone also coming all next year. We did efficacy and safety first, and then we're going to dive into all of that. It's coming next year. Team's been very busy preparing all of that, so you'll be seeing it at upcoming conferences.
Great. So as I transition to the next wave of questions, Larkin, do you want to just quickly introduce others from others from Gilead so that we can make sure that people know who's going to be answering questions?
Yeah, absolutely. I'm actually just going to call people out. So it's myself, Larkin, Global Community Engagement and Advocacy. We have Jeremy Schmalzley, who is our lenacapavir strategy lead. We have Igor from our contracts and legal department. We have Jared Baton, who many of you know, who heads up our clinical development programs. And we have Ryan McKeel, who is on our public affairs team for our intercontinental region.
Fantastic. Thank you for that. So a whole bunch of access related questions, and totally take your point, Asia. There's obviously way more to unpack across the board. We're going to get as far as we can. I want to start maybe in the process of access. First with regulatory, there were some questions both about planning regulatory filings for both FTAF for cisgender women, which was actually the genesis of Purpose One for those historians amongst us. So the FTAF plan, and then someone else asked a question, I forget who it was, about regulatory approvals and the process by which that's happening and the timelines. So does one of you want to take on?
I'll take that.
Question? Maybe I'll throw in on top of that, just pivoting back to the data forthcoming about pregnancy and breastfeeding, given the diversity in the trials, is the hope to get approval for LEN for 16 and 17-year-olds and for people who are pregnant and lactating all at once, or will that have to wait? So a bunch of regulatory stuff there.
We are very busy doing all the regulatory filing work now. Our goal is to file with the US by the end of this year, which means regulatory approval mid-year next year. Our goal is to file now with other regulatory agencies. They only have specific days that you can file. With the US, you can send it in at any time. So with other regulatory agencies, we have to figure out when they let us submit, and then we're going to be submitting. And we are also going to be submitting using, if approved, the EU Med for All pathway. We're still working through those details with the EU and the EMA. We're definitely filing in Europe and using the EU Med for All pathway. That ball is in their court, not ours. So we'll be ready to file by the end of this year in multiple different regulatory agencies, and we have to wait for the appropriate dates. We are also sharing our Descovy data with the US FDA also by the end of the year. And so all of that information, we're working as hard as we can. Some people thought that when we get the results, we're done. No, actually, when we get the results, the true work begins of all the regulatory filings all across the world. And this is going to be a really robust filing program with a true attempt of global simultaneous filings as close together as the regulatory agencies allow us to do.
Fantastic. I know, and maybe I'll just pick up on two questions just there in the chat about regulatory. I know a number of people in line from South Africa, lots of interest in SAPRA. I know it's been reported in the South African press about whether it's a SAPRA-specific application or through the EMA Medicines for All. Can you both clarify that from the South Africa perspective, but then picking up on Asia's question, the relationship between the 18 countries that you have set are priorities? Do you have a sense of how many of those are part of the potential Medicines for All program through EMA? How many of those aren't? And will those happen before the end of the year or into early next year?
The EU filing and EU Med for All filing will happen next year because of them. They have specific times that you're allowed to apply. So we have to apply with EU and EU Med for All next year. South Africa is definitely involved, but there's nothing firm yet on all the countries. We just met with all these regulatory agencies. We have regular meetings and correspondence with them, and we're still working through all the details. So I can't tell you anything official, but South Africa is our highest priority country in working with EU Med for All. And I believe SAFRA has said that they are working with EU Med for All publicly, so I can tell you that part. So that's one of our highest priorities. You saw the distribution of participants in Purpose 1, and we understand the incidents, so that's a big priority. For the rest of the questions that are veering more towards which countries and where, et cetera, I'm going to defer over to our colleagues who are experts on the access plans for those countries. I will just say the regulatory filings with the other agencies other than the FDA are all going to be happening early to mid to later next year based on when those agencies allow us to file. It's not us. If it was us, we would do it at the same time as FDA. All
right. And maybe before others jump in, there was a question that came in, I think, quite related to this from Jessica Burry about India. We know, obviously, it certainly was a big issue for CAB and regulatory approval and generic manufacturers there. Is there a regulatory strategy for India to help expedite those generic companies that are based in India?
I'm going to turn that over to our colleagues. Larkin, can you help say who should answer that question? Jeremy or Jared?
Jeremy or Igor, whichever one of you would like to jump in there, feel free, or Jared.
Yeah, I'm thinking, Igor, do you know how it works with India? Is Igor there?
I'm happy to jump in. Thanks, Jared Baton. Nice to see everybody. So thank you for the question on India, and thank you for the question on the additional 18 countries. We're taking in all of the opinions on how quickly we should be registering in each country. We're moving as expeditiously as possible. Thank you on India. It's one we have flagged already. We're trying to figure out how to expedite that one in parallel with everything else. For the other 18 countries, as Mopley said, part of the process with the EU for All is a discussion both with countries and with the EU process and with WHO in consultation on assembling a group of countries. That is a process that's not fully within our purview, but we are making advocacy for trying to get as many of the 18 countries into that process as possible, as well as simultaneously working on filings for those places. So thank you for pushing on all of those questions. Mitchell, back to you.
Thank you. So let me then move into the next set of kind of from regulatory into manufacturing, then we'll get to licensing. A question came up, and again, apologies, I don't remember all the names as I was making notes of a sequence here. A question about the manufacturing capacity, both from Gilead as the originator, and if you have a sense of the manufacturing capacity of the 6 companies that you have licensed already, and I'll get to the other licensing related questions. But let's first focus on manufacturing capacity.
Yeah. So I'll start on this, and I welcome colleagues to jump in. For Gilead's manufacturing capacity, our intent is absolutely to make sure that there is no gap in capacity during both for anywhere in the world that wants to be using lenacapavir and for these next period, particularly the period until generic manufacturers for low and lower middle income countries are able to rise to the millions of doses that I think the world would expect to be needed. So in the meantime, we are working on lines of manufacturing and capacity for being able to generate millions of doses for an interim time-limited period, hopefully, where we can accelerate generic manufacture as quickly as possible. I will tell you that the generic manufacturers we've already initiated technology transfer even before we have a fully approved agent for a fully approved label anywhere in the world. Thus, we are able to push out as much as we can for the manufacturing technology information for those countries. For the question of their manufacturing capacity, millions. The anticipation is that these are all quite capable, high-quality manufacturers who are able to produce end-to-end. And this was one of the pieces that we thought about in the choice of manufacturers. I did see there was a question on how choice of manufacturers was done. It was essential that they're able to do end-to-end, from the synthesis of lenacapavir itself, from the chemical synthesis of the active pharmaceutical ingredient, the API, all the way through the fill of a sterile injection and the ability to package a sterile injection. And all 6 of them are able to do that, and able to do that at the highest capacity. And I think we chose 6, recognizing that they are 6 and they are non-exclusive and more can be added. And we would love to hear, we would love to continue to hear advice on who else has the capacity to do this. But 6 was chosen so that there could be good competition, which could both elevate the numbers that are able to produce and to be able to drive down price in the generic market. Thank you.
Great. And let me just pick up on one question, I think, from Fatima. There is no South African manufacturer. There obviously are generic companies in South Africa, clearly a priority country, as you all stated. Can you give a little sense of why no South African company, and what might happen, and could you add?
Yeah. Absolutely can add. These are non-exclusive licenses. We would absolutely want to be able to work with. There is one company in Africa. And there are 3 countries. So it's 2 continents, 3 countries, 6 manufacturers. There's ability to add additional manufacturers. These are non-exclusive licenses. All of the manufacturers have the ability to do end-to-end, and that's one of the. I think it's not fully appropriate to talk about a specific manufacturer. But one of the reasons that some manufacturers are not part of the agreement right now is because they did not have the ability to do either the synthesis of lenacapavir cells. They don't make their own they don't do their own chemistry, or they don't do all the way to a sterile injectable finish. Obviously some manufacturers are quite expert at either buying active pharmaceutical product and making it into pills, or quite expert at making and making pills themselves, but not making sterile injectables.
Great. Thank you. And I know we are officially at the bottom of the hour. I know some people will likely have to jump. We will keep recording. I hope we can stay as many as possible for the next 15 minutes, because obviously there's a lot undone here. I think obviously the critical question in so many ways, and the science is amazing, but the volumes, how many manufacturers, it all depends on the price. I did want to just be clear. What can you say about the price generally, not just about low and middle income countries? If you can't give us a price on this call, when will that price be available? And because that is the determination for every donor and every government, obviously.
Yeah, and thank you. And it's truly welcome everyone's comments on this. Part of what we've been saying is that we've had lots of meetings with lots of people, and we've wanted lots of we want lots of advocacy. We do advocacy ourselves, and we want everyone. We want a variety of opinions. So I can't give you a price right now, and I can't give you a price not because I'm being coy, because we actually don't have a price right now for any setting, because we are actively working on what it takes to make that price the best for every setting. For low and lower middle income countries, I will tell you, even yesterday we were having discussions on how we can figure out the price that is going to allow a large volume, good initiation of lenacapavir for that time period before the generic manufacturers are able to start up, because we want them to be successful. We want countries to be successful. We want generic manufacturers to feel like there's reason for them to want to produce the millions that they have the capability to produce. And we're working actively on how to make that to make that access price as best as possible. And
when will your price be announced, Jared?
Hey, Asia, thank you for the question. I don't have a date right now. We will be happy.
When will you have a date?
I don't have a date for a date yet, but we will happy to give everyone a heads up when we have that information. Thanks.
One other piece in addition to price, I mean, you mentioned low and lower middle income countries, very clearly a key part of the strategy related to the 18 countries initially and to the licensing agreements. It was just announced, just as people were descending on Lima, Peru, a host country of not only the Research for Prevention Conference, but obviously a host country of Purpose 2. A lot of questions in the chat, and you know them. What about Latin America? Not only the countries that hosted Purpose 2, but Latin America generally and Asia, regions of the world that are actually seeing increasing infections, even as sub-Saharan Africa Southern and East Africa are actually seeing declining rates of new infections. So what's the strategy for Latin America and Asia, both countries that hosted P2 and otherwise?
Thank you. And yeah, this is an excellent question, and I'm going to try to integrate. I've been trying to read the Q&A. I'll try to integrate some of the sub questions that are within that. So you're right, Mitchell. So within hours after the announcement of the Purpose 2 results, as promised when we had the Purpose 1 results, we said we would be able to talk about voluntary licensing. And so the voluntary license agreement for lower middle income countries of high incidence, we tried to be thoughtful in the selection of where incidence is high and where World Bank classifications of low and lower middle income. That 120 countries, I think to our knowledge, is the largest and earliest voluntary license agreement for an HIV drug that's been done. And 120 countries is not all countries in the world. For Latin American countries, including, for example, Brazil and other Purpose 2 countries, Gilead has, for a long time, felt very committed and has persons on the ground in those countries to be able and has worked through the ability to produce medicines for those regions. And currently, there are tens of thousands of people every day who take Gilead HIV treatment medicines in Mexico, in Peru, in Brazil, in Argentina. And the plan for Latin America, as for other upper middle income countries, is working with governments, tiered pricing, dedicated manufacturing, and other creative solutions to be able to figure out what is right for those settings. Any of my other colleagues, feel free to jump in too as well, if I'm forgetting something.
Jared, you said most of it. I would just say that, voluntary licensing is just one component to an comprehensive access strategy. And so we are working through these. I get the sense of urgency and wanting to know as quickly as possible. And the second we have something, we will definitely tell you. But there are ways that we can do it. Some people have mentioned tier pricing. Yes, that's one component. But there are also other innovative strategies we can do for these countries to have efficient and sustainable access. And so that's what we're working on. We're working on registering in them, working on making sure we have all the manufacturing and the appropriate volume needed for demand. So rest assured that we are not just announce the voluntary licensing and then walk away. We are very committed to making sure all these countries have access.
Thank you, Jeremy. I think to reinforce that, I think we want to continue to convey that we're committed moment to moment as we are on this conversation right now, and we're committed we're committed for the long haul. And since June, when we first had results, we have said we will tell you as soon as it is multiple pieces that are going to make this truth. And we will tell you as soon as we have information on multiple pieces. So, for example, we said in June, we are going to move to voluntary licensing. And we couldn't tell you exactly how we were going to move to voluntary license or which voluntary licensees in June, but the moment we could in September, we did. And so we commit to continuing to say we are going to keep fulfilling, and there are going to be many more mileposts along the way, and we will tell you as soon as we have each little bit in place. And all of those we hopefully add up to the kind of access and impact that we all want.
Joe, thank you for that. And I do want to pick up because I know Kenneth Mohanga asked earlier, and I know it's back. Asia's asking again about particularly the purpose 2 countries that are excluded, and the rationale, the justification, and the plan for that.
I'm sorry, Mitchell, it cut out on my side. Can you say it one more time? I'm really sorry.
About the four purpose 2 countries that are not part of the agreements, and how that fits in. Kenneth had asked early on in the Q&A about what the access plan is for those 4 countries. Asia's asking about why those 4 were actually excluded from the licensing, given their contribution to the studies.
Yeah. No, I think so. Thank you for the question, and I'll say the commitment Gilead has is that we view lenacapavir as potentially making an impact in every country in the world, and so we are working through. And it is a challenge to say, but each country has its own prioritization of HIV prevention, its own prioritization of health, and its own ability to achieve access. And so we're working through each of those on its own. For the 4 countries in Purpose 2 in Latin America, as well as the United States, and I think that's also the United States, we working through individual plans for those countries for what access looks like. Every person who's been in Purpose 2 will continue to receive lenacapavir should they want, until there is regulatory approval and access availability for them in their country, whether that be here in the United States or in Sao Paulo. And then for Latin America, it is exactly what we've already done before for access to HIV medicines, for example, which includes dedicated manufacturing and pricing that works for Brazil or pricing that works for Argentina, and then working directly with governments to figure out how prioritization of prevention and prioritization of access can actually be achieved.
Thank you all. Obviously, there are questions and comments that are not fully addressed, and we knew going into this that there's no way that an hour or a 75-minute phone call would ever cover it all. Absolutely agree that there is a need for multiple follow-up conversations on a range of issues. I do, though, want to make sure we have time just to turn it over to my colleagues, Sarah George Carballo and Joe Radovosian, to just quickly talk about a document. As many of you know, much of this sits with Gilead. It is their product. We need it to be approved, and that means they need to file it. We need it manufactured. We need an affordable price. All of that is a given. And we need governments and donors to be able to work with it. And that obviously requires an affordable price. It also requires a number of other aspects. And so shortly after both Purpose 1 and Purpose 2 were out, we created the Lens on LEN just to make sure people had the background on the actual product and the trial results. In October, we put out a plan that looks comprehensively at all the moving parts about introducing LEN and how to accelerate that so we don't squander the opportunities as we have with oral PrEP and cabotegravir and the vaginal ring over the last 12 years. And just yesterday put out a new document. So I'm going to quickly turn it over. Please don't leave yet, anybody. We'll do a quick wrap up, but I do want to turn it over to Gerard and Syra just to very quickly describe what's in that document and why it outlines the work that needs to happen, not only with Gilead, but with a number of other stakeholders.
All right, sure. Hi, everybody. I'm happy to go. And I think we have a couple quick slides for everybody. Thanks to the Gilead team for the presentation today. As folks know, I work at both Yale University and Duke University, but I was a consultant to AVAC on this report, and I had the pleasure of working with Syra while we were on the entire AVAC team on this. You know, this past week we celebrated a lot about the increasing number of people that have been put on PrEP over the last year, but we all know that it's far from the reach that we all need to have transformational impact on HIV incidence and ultimately change the trajectory of the epidemic. This is what this report is meant to do, is to highlight what it needs to take to get to these ambitious goals. So what we did is we outlined the different gears, what we're calling gears that are related to lenacapavir rollout, and we focus exclusively on lenacapavir for this, emphasizing the different roles of diverse stakeholders that play. We heard a lot today from just one of the many different stakeholders that are involved in this. We include everyone that's involved in the journey, including product discovery, development, manufacturing, procurement, policymaking, regulatory, implementation, science, as well as on the delivery aspects of it. As you can see in the infographic here, that we've got national governments. Think of these as ministries of finance, ministries of health, broad political leadership at the highest levels that help set the tone and drive the policy and programmatic adoption at the country level. Of course, we have donors and procurers as another big stakeholder. Or these are organizations like PEPFAR, Global Fund, Gates Foundation, SIF, and others that negotiate pricing, set procurement agreements, they enable access, and they also drive program implementation and provide financial support. At the center of all of it is civil society. These are advocacy groups, community organizations, many who are on the call here, that help create demand generation, educate communities and providers, and hold all of us as stakeholders accountable. We've already heard a lot today from Gilead, which is responsible for setting pricing, ensuring product capacity, facilitating regulatory approvals, collaborating with stakeholders for an effective rollout strategy. We wanted to highlight Gilead license holders because Gilead was quick to announce to 6 pharmaceutical companies non-exclusive agreements that are licensed to provide generics in 120 countries. And so we consider them to be an early stakeholder that needs to be engaged at this early stage. And then we have, of course, international actors. Think of these as WHO, UNAIDS, UNITAID, and others that provide technical guidance, funding, and also shape the implementation environment in which a product is introduced. Like interconnected gears, when one turns, others follow, demonstrating how coordinated strategic collaboration can leverage our collective strengths to help overcome the barriers that we're accustomed to over previous PrEP rollouts. That's the idea behind this. I'll walk you quickly through just the next slide, and then you can see all the details in the report. The roadmap is organized around 3 phases, time phases of rollout. Phase 1 is when we are preparing for product availability. That is now. That's phase 0. Phase 1 is defined as a time when the originator product, which is Gilead, becomes available. And Gilead has indicated that they have an 18-month manufacturing lead time. And then phase 1, as we're defining, is the period where generics come online and produce generics that are available for broad program implementation. We lay out, as you can see, a number of action steps across each of the phases. I'm not going to read these out. You have the details in the report. But just to highlight at the top line, phase 0, the phase that we're in now, is the foundation. This is where foundational preparations are underway, and it's arguably the most important phase, as many people have said. This is the phase that sets the tempo and it sets the ambition for what follows after. This is when we need transparent and affordable pricing, financial frameworks emerge during this phase. Donors have to signal demand that they need for procurement. Governments need to prepare ambitious rollout plans. Civil society needs to be positioned to support demand generation and ensure accountability across different stakeholders. WHO developing guidelines, with a regulatory approval expected next year, et cetera, et cetera. And then phase 1, you'll see a number of different actions that kind of shift the focus more towards national level implementation, registration, distribution, supply chain strengthening. Donors and procurers need to continue to be active in this phase to provide financial support for rollout, for community mobilization, for civil society activities. Innovative financing and public-private partnerships are critical at this stage to ensure that sustainability of these programs continue. Phase 2 is when we transition to generics. There's a recognition that there'll be a mix of both branded lenacapavir and generics available for governments to be deciding on programs. We'll continue to need innovative financing mechanisms, public-private partnerships to increase financing, and of course, international partners need to continue to develop a research agenda and other efforts that are needed for sustainability and financing. So all of these are highlighted in the report. We hope that it's helpful for planning purposes for all different stakeholders. I just wanted to also quickly share that this is a product of several interviews that we conducted over the last few months. We also held a workshop at R4P. People were very forthcoming in terms of their frustrations, their recommendations, their ideas for how to incorporate lessons learned based on oral prep, based on early experiences with cabotegravir, as well as the ring. And so we hope this is a helpful roadmap, and I know AVAC and others will create more opportunities to have conversations about it in the days ahead. So thanks so much. Back over to you, Mitchell.
Great, Gerard, thank you so much, and I really appreciate that. And the link is there. And I obviously this is the first of many conversations. The idea was not to mute anybody; it was to try to get as many questions in. And I'm sorry if people feel that way. I'm happy. Asia, you've raised it. I'm happy to have you come off mute and add to what's in already in the chat and in the Q&A. This is one of many conversations that need to happen. It's not to resolve it all. It is to get a grounding in where we are and clearly where we need to go. And there are many, many, many things that need to happen. And Asia, I'm absolutely happy to have you come back off mute and provide any input.
Thanks. I think the concern is from more than 100 people on this call that fundamentally excluding territories where communities put their bodies on the line and participated in the trials that generated a massive bump in your profit shares are then being excluded from your access strategy is unacceptable. And we want that changed. And in the meantime, we want the anti-diversion clauses removed so that if those countries, Argentina, Peru, Mexico, Brazil, issue compulsory licenses, they can actually satisfy them with products from those licensees and do something about the pace of scale-up that Gerard just described. We find it intolerable to imagine another 3 years where we're accepting the status quo and accepting the pace that Gilead has set. We need to explore aggressively every single opportunity to continue to collapse the time to introduction. And it's simply unethical that we're emerging from a starting point where we're hearing about diversity in clinical trials and a refusal to allow countries to participate in an access strategy. It's a clear violation of the Helsinki Declaration. And we've been saying that since Munich, and your responses haven't changed. So we're extremely frustrated. It feels like complete disregard for the concerns and the demands backed by evidence that you've seen in the chat from activists today, and you'll continue to hear. We need answers, Gilead. We need answers.
Asia, thank you. And we will, and I think to Andy's point in the chat, we will absolutely be sharing the recording online for all, and as well as the questions, and we'll provide those to Gilead, and they can provide any additional responses to those. But Jared, Larkin, Jeremy, anyone want to respond?
I want to say thank you, and please continue to tell us how to do this work to do this work together with everyone. And thank you for those who are continuing to comment. We want nothing more than to have lenacapavir, should it be approved, I have to say that, should it be approved in countries all over the world, be able to achieve the impact that we all want, because the status quo of 1.3 million new infections a year is also unacceptable to us. We want to work with you, with governments, with scientists, with clinicians, with other advocates all around the world to figure out what works best in every single setting. And we will continue to engage. We will continue to tell you what we're going to do and what we're able to do, and to try to project what we're going to try to do next. And we're going to edit as we get input on what's working, what's not working, and how we can achieve that impact that we want to get to together. Because we would like nothing more than to not have to work on HIV. And if we can all get there in some way, we know we have to do that together, because we know we can't do this alone. Thank you to Gerard for laying out that it really is an ecosystem, or an ecosystem of gears that's going to get us there. And thank you for everybody who's continuing to tell us all of the pieces that we can do this better. We will say not every decision that we're going to be able to make will satisfy everyone, because we certainly have contradictory opinions of what we're doing, but we are aiming for impact from lenacapavir. We're aiming for, really, if we can, contributing our part with everybody else to ending this epidemic. So thank you, really thank you. And we take everything in that you are saying.
Great. I want to thank everybody. And it is that ecosystem and absolutely demands on Gilead to answer these questions, to give us a price, to make that price affordable. You know that. Demands on PEPFAR and Global Fund, some of whom are on the line, and ministries of health who are also on the line to procure this product and be clear what the volumes they can absorb and program. And it's on all of us as advocates to continue to push. And we know that it didn't happen with cabotegravir. It didn't happen with the vaginal ring, and it didn't happen with oral prep. And oral prep did not have a constrained supply. And although people said it was too expensive 12 years ago at $54 a year, it's now seen as the new normal, the new baseline. We have failed as a global community, and I know we might disagree on different points, but I think we could all agree that we have gone way too slowly for 12 years, and I certainly don't want to be here 12 years from now and say, oh, wow, I wish we had. We know what we need to do, and we all need to be moving in those directions. And I really appreciate everybody's input in the chat, in the Q&A, on the line, and know that this is not a conversation that ends on December 3rd at 6 p.m. in Geneva, but rather one that really needs to get accelerated. And we will work with anyone and everyone to cultivate as many conversations globally, regionally, and nationally, and at community level as are needed to make that happen. And that is a commitment certainly that you have from me to the degree that we can make that possible. So I want to thank everybody. We will post the recording. These documents are there. If they miss things, if there are other actions that need to happen from Gilead, from civil society, from governments, from donors, name them. The more we name them together, the more we push and pull in the same direction, the better. So really want to thank you all for all of that. And I look forward to actually getting people on all forms of prevention and seeing far fewer than 1.3 million new infections in 2025, because I'm really tired of this. So thank you all, and we will post all of this, and we will definitely be in touch. Cheers all.
Thank you.